I'd like to say thanks to the committee for this award. It's absolutely fantastic and I'm honored, so thank you very much. So I'm going to see if I can get this working. All right, I have no conflict of interest at the moment. I'm hoping that's going to change in a couple of months, and I'll tell you why. But at this particular point in time, I have zero conflicts of interest. So we are interested in hormone-dependent cancer, so mostly breast and more recently prostate. And the reason should be pretty obvious. So let's use this. Oh, there we go. These are very common diseases. So in breast cancer, there are about 50,000 new cases in the UK alone every year. And a full 3/4 of these are driven by a transcription factor pathway called estrogen receptor. And then, of course, we know that we can block this. We have a number of different agents that are used to target estrogen receptor positive disease. And they come in different shapes and different flavors, and they essentially all do the same thing, though, and that is they block estrogen receptor transcription activity. And these drugs include selective estrogen receptor modulators like tamoxifen, aromatase inhibitors, and then pure steroidal antiestrogens. And essentially, as I said, they all block the ER transcriptional pathway. Now, we know for many women, these drugs are essentially curative, but we also know that a large fraction of women will develop endocrine resistance and die. Now, trying to identify which women are likely to respond to the treatments we have and which women are not going to respond is a big business, and it's quite a complex issue, and we really haven't resolved it yet. There are a number of different assays out there that will predict whether a woman's likely to respond to an endocrine agent or not. They all work to differing degrees, but actually there's been a biomarker that's been around for decades now called progesterone receptor. And I'm going to talk about this quite a bit. It's really fallen out of fashion recently, but I think it's an interesting protein, and we've pulled it out of a screen. And we've been very interested in this because we think it reveals new insight into how estrogen receptor works, and it reveals new therapeutic opportunity. I'm sorry, this pointer is not working very reliably. Here we go. So what we do know is that ER positive, PR positive patients tend to have a better outcome. Now that is a generalization, but that does tend to be the case. And women that have a disease that is estrogen receptor positive and progesterone receptor negative tend to have a worse outcome. Now this is, I guess, the simplest way of looking at this pathway. So estrogen receptor is a transcription factor. It needs some friends, so it recruits a number of cofactors. These cofactors have enzymatic activity and allow this complex to modulate chromatin. And essentially, this complex will bind to DNA and activate gene transcription events. And these transcription events culminate in cell cycle progression and cell growth. Now, one of the genes that estrogen receptor regulates is the progesterone receptor gene, and the link between ER and PR was really limited to PR being a biomarker of estrogen receptor activity. So given the fact that PR is a target gene, it had been used for many, many years as an indicator of whether a tumor is likely to be driven by a functional estrogen receptor complex. So essentially, if PR is there, ER is working, and drugs that block ER are likely to work. So really, these are the key questions in my lab, and they're really the how and the where. So where in the genome does estrogen receptor make contact? So these are the cis-regulatory elements that estrogen receptor will use to then regulate gene expression. And we've been very interested for a number of years now in how estrogen receptor makes contact with the chromatin or DNA. Now, if you look at the old textbooks, it looks pretty straightforward. Estrogen receptor will come in, bind to a responsive element, recruit some cofactors, and gene expression will occur. But we know it's not that straightforward. For estrogen receptor to be able to access DNA, it needs proteins that help open the DNA and essentially dictate where in the genome estrogen receptor can and cannot interact with. And we've been very interested in trying to identify these proteins. And ultimately, we've been very interested in trying to identify commonalities and differences between drug-sensitive and drug-resistant contexts, really with the goal of trying to identify key components or hubs in the ER complex that might be therapeutic targets. So a number of years ago now, we went about trying to identify in an unbiased manner where estrogen receptor interacts with the genome. Now, up until that point, we'd been studying the promoters of one or two target genes, and we were pulling those promoters apart in fantastic detail and making pretty big conclusions based on one or two promoters. And then the technology came along that allowed us to interrogate the whole genome. We coupled it with the chromatin IP, which is a way of pulling out estrogen receptor-bound DNA. And we found almost overnight that actually estrogen receptor very rarely binds to promoter regions. It regulates genes from quite significant distances. So it will associate with, I'm sorry, this pointer is not working very well. It will associate with these distal regulatory elements, and then there are chromatin loops between the regulatory elements and then the target genes. And what we could do when we mapped estrogen receptor binding sites was we could put the thousands and thousands of contact sites together and look for patterns. And essentially what we were looking for were DNA sequences that were overrepresented. And we found this sequence here. And it meant absolutely nothing to me at the time, but when we ran it through the database, it told us that this particular sequence is bound by a family of transcription factors called fork-head proteins. And so we became very interested in fork-head proteins, and it turned out there's a specific fork-head protein called FOXA1. And these two proteins, that is estrogen receptor and FOXA1, work in unison. So estrogen receptor, we know, is the driving nuclear receptor, but it looks like it requires what's called a pioneer factor to be able to access this compacted chromatin. And that pioneer factor for estrogen receptor is FOXA1. So FOXA1 is essentially it's a tethering mechanism or an adapter that allows the estrogen receptor to make contact with compacted DNA. And then this entire complex then loops over and regulates target genes. Now, we spent a number of years trying to tease apart the role of FOXA1, and it looks like it's absolutely critical for estrogen receptor function. So estrogen receptor will not bind to chromatin on its own. Now, importantly, it will bind to DNA that's not chromatinized. So if you take a piece of DNA and you put it in a tube and you add estrogen receptor will bind to the DNA and it will elicit gene expression activity. But if you take that same sequence, that same piece of DNA, and you wrap it around histones, estrogen receptor cannot bind. It needs pioneer factors to open the chromatin. And FOXA1 plays that role. So these two proteins, in conjunction with a third protein called GATA3, which I'll touch on right at the end, look like they are the core components of the estrogen receptor complex. So ER, FOXA1, and GATA3 together are three critical transcription factors that allow estrogen receptor to be a transcriptionally active, hormone-responsive nuclear receptor. If we get rid of FOXA1, estrogen receptor doesn't bind to DNA. So we and others have done this numerous times now. In the absence of FOXA1, ER is inert. It simply can't bind to the chromatin. We think it's still there, and in fact, we still think it can interact with cofactors. It just can't bind to the chromatin. And as you would expect, you don't get gene transcription in the absence of FOXA1, and you don't get cell division. So FOXA1 really is critical for estrogen receptor to function. Now, we and pretty much every lab in our field have been trying to identify escape mechanisms or mechanisms that contribute to endocrine resistance, and there are many of them. And they come in different varieties. So we know that growth factor pathways can be amplified. We know there are copy number changes in important cofactors. And more recently, we know that estrogen receptor itself can be mutated. And I have to say, this was a little bit of a surprise because we've been sequencing estrogen receptor for decades. And according to everyone in the field, estrogen receptor was wild type. Right? It didn't get mutated. But if you look in the relapse context or the metastatic context, it is heavily mutated. And the mutations are in very, very predictable points. It's usually amino acids 536, 537, or 538. And we had missed this for decades because we were sequencing primary tumors instead of the Mets. So it looks like ER is getting mutated. We're still trying to get a handle on the frequency. It's probably going to be somewhere around about 20 or 25%. But the mutations are in very predictable residues. Now, regardless of the escape mechanism, FOXA1 is dependent. So we've been accumulating as many resistant models as we can. Mutant estrogen receptor, amplification of growth factor pathways, amplification of AIB1 and associated cofactors. And in all contexts, they are endocrine resistant. And in all contexts, they are dependent on FOXA1. So if you block FOXA1, you stop growth of endocrine resistant models. Now, more recently, data has come out from Newcastle showing that in castrate-resistant prostate cancer models, FOXA1 is also dependent. So I guess the analogies are quite high between mutant estrogen receptor and splice variants in androgen receptor, which we see in castrate-resistant prostate cancer. These mechanisms bypass endocrine activity, so they're essentially mechanisms to bypass drug responsiveness. But in all cases, the nuclear receptor gets switched on, and it still needs its pioneer factor to be able to bind to the chromatin. So we've tried to identify ways of regulating FOXA1. Now, it's a tricky protein. We don't know what it looks like. So people have tried to crystallize it and they've failed. It's a very tricky protein to purify. So we don't know what it looks like. It's a transcription factor, which are typically considered undruggable, other than the nuclear receptors, of course. But we went about setting up assays that would allow us to screen for compounds that would block FOXA1. Not necessarily directly, but potentially indirectly. So we teamed up with Cancer Research Technology, and we screened their compound library of about 180,000 compounds, and we got a hit, and this hit, it turns out, regulates a kinase. And it was an unknown kinase. And so we went and did the kinome screen and screened all human kinases to find the kinases that were responsible for FOXA1 activity, and these two projects converged on the exact same kinase. And so we're pursuing this kinase at the moment. It is relatively unknown. There's only a handful of papers on it, but this kinase is only expressed in hormone-dependent cancers, and it looks like it's only dependent or only required in hormone-dependent cancers. Now, we took this as far as we could. This is really not the remit of our academic lab, so we've had to team up with industrial partners to be able to pursue this, and I'm hoping in a year from now that I will have a conflict of interest if this is successful. So time will tell. Now, one thing we do know about estrogen receptor and FOXA1 is that the relationship is dynamic. So estrogen receptor and FOXA1 can physically interact, but where in the genome they go can change. And we identified this when we went about mapping ER binding sites using our method called ChIP-seq in primary tumors. So up until a few years ago, we'd been studying estrogen receptor and FOXA1 in cell line models growing on plastic. And then we established these methods in pieces of primary clinical material, really with the goal of being able to prove that the conclusions and the paradigms we were making in the cell lines were accurate, but also to be able to study estrogen receptor binding profiles in tumors that had different outcome, which we'd not been able to do up until that point due to the limitations in our cell lines that were available to us. So I had a really great PhD student, Carroll, who went about setting up ChIP-seq in clinical samples. These are intact pieces of tumor, so we haven't separated cells or sorted cells in any way. And she mapped estrogen receptor binding by ChIP-seq, and it worked really, really well. And what she could do was she could compare the ER binding profile in tumors that had an ER positive, PR positive IHC profile. And we know these women had a good outcome. And we could compare that with the ER binding profile from tumors from women that had a bad outcome and died of breast cancer. And as we expected, these tumors had the hallmarks of a poor response in that they were ER positive but progesterone receptor negative. And once we stabilized the data and we started to look at it, it was pretty clear that the estrogen receptor binding profile changes. So ER can bind to one set of binding sites in good outcome tumors, but actually occupies a different set of regulatory elements in women that are going to relapse on endocrine agents. Now, what was interesting was even though estrogen receptor moved around the genome, it looked like it went from one subset of FOXA1 binding sites to a different subset of FOXA1 binding sites. So it's not random. When estrogen receptor moves around the genome, it doesn't move randomly. It goes from one set of pioneer sites to a different set of pioneer sites. Now, this has really been the million-dollar question for us. Why does estrogen receptor move, and what are the mechanisms? Now, sorry, I forgot this slide. I'm going to come back to this in a little bit, but I just want to point out that when we crunched the numbers, we actually had about 600 of these good binding sites, these good outcome ER binding sites, and about 1,200 of what we consider poor outcome binding sites. So these are the binding profiles that we identified from the primary tumors. And I'll come back to this in a few slides. So we've been very interested in trying to identify what the mechanisms are that allow estrogen receptor to move around the genome, and there are a number of working hypotheses. So one is that there's, and I think they're probably all right to certain degrees, one is that there are post-translational modifications on ER or FOXA1 or histone modifications or some of the cofactors, and then this changes where in the genome estrogen receptor can bind. Another possibility is that it's just changes in chromatin structure. So maybe there are subtle changes in the opening or the closing of chromatin, and this allows estrogen receptor to preferentially bind to one set of sites versus another. Another possibility is that there are differences in associated proteins. Now, we actually explored this last hypothesis first by setting up a modified chromatin IP. So it's a method called RIME, Rapid IP Mass Spec of Endogenous Protein. And essentially, it's like a chip. So we cross-link our cells. We pull out our protein of interest, and instead of looking at the DNA, which we've been doing for years with our ChIP-seq approaches, we were looking at proteins using mass spec. And it's a discovery tool for identifying interactomes or finding interacting proteins. And when we applied this to estrogen receptor, we pulled out most things that we had expected. So we pulled out all the usual suspects, GATA3, FOXA1, a lot of the cofactors. And there were a couple of surprises, but not a lot. It was mostly things that the field had pieced together over decades of good biochemistry and molecular biology. But what we found was if we actually stimulated our cells, and this was a bit of a side project, and actually that turned out to be quite interesting. If we stimulated our cells with both estrogen and progesterone, we found that progesterone receptor interacted with estrogen receptor. So these two nuclear receptors converged on the chromatin, where PR got recruited to the estrogen receptor complex. And I guess this isn't super surprising given the fact that these nuclear receptors share cofactors. We know that they have very similar DNA binding sequences. So I guess there's no surprise that these nuclear receptors can converge, but we pull PR out from an unbiased screen as an interacting protein. And this suggested to us that maybe PR is actually playing a role in the ER complex. So we went about testing that. So we performed a series of ChIP-seq experiments. And I'm sorry, this is a very busy slide, but actually if you just look at the example on the left-hand side, I think this epitomizes what we're trying to say here. So this is a ChIP-seq experiment where we've mapped globally transcription factor binding. And the top 3 panels, if my pointer will actually get there, I'm sorry. The top 3 panels are estrogen receptor ChIP, the next 3 are progesterone receptor, and then the bottom 3 are a cofactor called P300. And what we can see is these are all in estrogen-rich conditions. So we grow our cells with lots of estrogen. ER is all over the genome, but it's not at this site. But if we add either progesterone or a synthetic progestin called R5020, we see that estrogen receptor gets reprogrammed, or it starts to bind to these 2 particular regions here. And we see that this binding is coincident with PR binding and with P300 binding. And the fact that P300 is there suggests that that binding site is transcriptionally active. It's going to be transcribing something. Maybe a non-coding RNA, maybe it's going to be this gene down here, which I can't point to because my pointer's not working, but the one at the bottom of the screen, or maybe it's going to be a gene that's a megabase away. And if we actually crunch the numbers, it turns out that progesterone will reprogram ER to about 14,000 sites reproducibly in this particular cell model. And you can see by the intensity of this line here, we can also see coincident PR and P300 binding at all of these sites. So, and this is a pretty short pulse of progesterone, so it's about 3 hours. So if we grow our cells in estrogen and we give them a pulse of progesterone for 3 hours, estrogen receptor gets reprogrammed. So essentially progesterone will tell estrogen receptor where in the genome to bind. And so we got very, very interested in this, and we started to present this data, and we hit a bit of a wall in the field because if you read the literature, progesterone is bad. Right? Much of the literature says that progesterone is pro-proliferative, and it's not something that you want to be looking at. We are looking to stimulate cells with progesterone, potentially to modulate estrogen receptor in a therapeutic way. But the literature would suggest that actually that would be a bad approach because the data from the normal mammary gland field suggests that progesterone is pro-proliferative, and that is absolutely accurate. So we know that in the normal mammary gland, both mouse, and we can glean insight from the hormone replacement trial data, in normal healthy tissue, whether it's murine or human, progesterone can be pro-proliferative. It's only in a very small subset of cells, but progesterone can be pro-proliferative. And extrapolations were made from the normal mammary gland to cancer, and the conclusion was that progesterone is bad because progesterone will increase proliferation, which is the last thing you want to do if you're trying to block cancer. But it turns out that actually in cancer, things are flipped. So we know that progesterone receptor expression profiles, that is the cellular expression profile of progesterone receptor, is different between normal and cancer. We know that target genes are different, and we know that the functions are very, very different. And it turns out, of course, that progesterone is anti-proliferative. It is a therapeutic that is used in ER-positive breast cancer. So there is a fundamental rewiring of the pathways that occur in normal mammary gland biology relative to cancer. And for many, many years, progesterone was largely ignored because of this extrapolation. Now, I think this is inaccurate, and it's something that we really need to go and revisit. So we've tried to add data to the confusion. So we've tried to look in every single system we could, in vitro, ex vivo. We've gone back, we're doing a clinical trial, which I'll talk about in a second, to assess whether in ER-positive breast cancer context, whether progesterone is proliferative or anti-proliferative. And in all cases, it looks like it's anti-proliferative, which actually matches with the cancer data. So these are tumor xenografts. We've done this for all the cell lines that we can get our hands on. If we grow them in the presence of estrogen, they grow very, very nicely. If we co-treat with progesterone, we see this diminution. It's not a complete inhibition, but we see about a 50% reduction in tumor growth in the presence of progesterone, which would imply that progesterone is actually anti-proliferative and can block estrogen-mediated tumor growth. We've taken a number of these cancers and we've mapped ER binding by ChIP-seq, and we see the exact same thing that we see in the cell lines. So we see this global reprogramming of estrogen receptor binding in the presence of progesterone. Now, we've also tried doing this on clinical samples. So we set up, in collaboration with Wayne Tilley from Adelaide, we set up an explant system that allows us to take pieces of tumor straight from surgery, and we can cut them into little pieces and randomize them and essentially do an ex vivo trial where we can take pieces of the same tumor and grow them on these special sponges and treat them with hormones and compare the proliferative response. And we're looking at, we've done a number of things with these tumors. We've looked at BRDU incorporation, but we've also looked at Ki67, which is a surrogate of proliferation. So essentially, if a cell expresses Ki67, it is proliferating. And we've done triple immunofluorescence here. And so we've done nothing to these pieces of cancer. Again, we haven't broken up the cells. We've just taken them from surgery, cut them into little bits, randomized them, and we've treated them with either vehicle, estrogen, progesterone or estrogen plus progesterone. And really, if you look at this second and the fourth panel, I think the point's pretty clear. So if we add estrogen, we see an increase in the number of blue cells. So these are the proliferating cells. And if we co-treat with progesterone, we see that decreases. And again, we've taken these explants and we've performed ER chip sequencing on them, and we see the same thing that we saw on the cell lines in plastic and the same thing that we saw on the cell lines in xenografts. And that is that by treating these pieces of primary tumor with progesterone, we can push estrogen receptor around the genome. So we can cause a really global reprogramming of estrogen receptor binding sites. And it's not subtle. You can see that very clearly. In these pieces of clinical material, if we co-treat with progesterone, we see 15,000 new ER binding sites. And these are sites that we see in multiple tumors and, of course, in multiple technical replicates. Now, interestingly enough, or maybe interestingly enough, if I can make the slide go forward. Sorry, there's a bit of animation here. Dave, is there a way we can make this? Oh, there we go. Thank you. So if we actually look at these progesterone induced ER binding sites, they encapsulate those good outcome sites that I talked about at the beginning. So remember when we mapped ER binding in tumors from good and poor outcome, we'd never seen these good outcome sites before until we did this experiment. And if we take these pieces of tumor and we treat them with progesterone, we can force estrogen receptor to go to a different set of regulatory elements that essentially represent those sites that we saw in the good outcome tumors. So we think actually what we've done in a dish is we've essentially recapitulated the premenopausal conditions. Now we've taken this one step further, and we've tried looking at the combination of progesterone with an estrogen receptor antagonist. And so we've grown all our xenograft tumors. We're doing this with explants now, as you can imagine. We've grown our xenograft tumors in the presence of estrogen, so everything is in the presence of estrogen. The xenografts grow quite nicely, as we would expect. If we add progesterone alone, we see this partial inhibition, as we've seen time and time again. If we add tamoxifen, the estrogen receptor antagonist, we see inhibition. But if we add both tamoxifen and progesterone, we see the greatest inhibition. And we saw this in all of the xenograft models that we looked at. So essentially, we're adding a PR agonist in combination with an estrogen receptor antagonist. And we're seeing the greatest inhibitory effects. Sorry, Dave, one more. Yep, there we go. So based on this data, we're actually initiating a window trial, and we've raised the funds for this, and we have a clinical fellow who's about to run it. And essentially, it is an extension of what I'm showing in this preclinical model here. It's combining an estrogen receptor antagonist with or without progesterone. Now, we're doing low dose and high dose, and we can talk about that later if people are interested in. Now, the reason we're interested in this is progesterone is cheap. Right? It's off patent and it works. It's still given by oncologists usually in very advanced disease. And in fact, the old clinical trial data showed that progesterone was as effective as tamoxifen when compared head to head. But it's usually used as a last resort now. What we're saying is we want to combine it with an estrogen receptor antagonist up front. Now, importantly, a number of women who are taking estrogen receptor antagonists, particularly aromatase inhibitors, will come off their treatment. And it's because of side effects associated with the drugs. And one of the major side effects is hot flushes. Now, actually, one of the best things for combating hot flushes, well, the best thing is estrogen. You're not going to fuel an estrogen receptor positive tumor with estrogen. But actually, what works really well is megestrol acetate, which is a synthetic progestin, and is not used because people are worried about the perception of progesterone being pro-proliferative, and it's not. So actually, we have a really good agent that can be used to combat hot flushes and can improve treatment compliance. And this is something that we're pursuing. So this is where we are at the moment. There's clearly this crosstalk between PR and ER. It's not that straightforward, of course. Mother Nature doesn't make things as straightforward. We know that there's at least one other nuclear receptor. Actually, we know there's at least two, because glucocorticoid receptor is also involved. So there's clearly communication between these different nuclear receptors. Wayne Tilley's been looking at the crosstalk between AR and ER. We've been looking at the crosstalk between PR and ER, but there's almost certainly going to be crosstalk between AR and PR, because these three nuclear receptors tend to be co-expressed in ER-positive breast cancer, and there is going to be communication between them, and we can exploit this information therapeutically. We just need to understand the biology to know what ligands to use and which patients to give it to. Now, underlying all of this are these, what we call pioneer factors. So these are really the foundation proteins that open the chromatin and allow these nuclear receptors to bind. I've talked about FOXA1, but in the last 2 minutes, sorry, Karen, I'm only going to read 2 minutes. I just want to show you some data on a protein called GATA3. Now, GATA3 is an absolute pain in the butt. Now, we've been looking at this protein for about 10 years now, and it is a challenging protein. It does a bit of everything, and it's very context dependent. It can be pro-proliferative, it can be anti-proliferative, it can induce metastases, it can block metastases. It is challenging, and it's been a source of much frustration for my PhD students and postdocs and myself. but it's an important protein and we have to figure it out. And one of the reasons we've been very interested in this is because it is heavily mutated in breast cancer. I'm going to show you just 2 slides of data. We still don't know what it's doing, but it's interesting, and I wanted to put it out there just for discussion. Now, if you look in primary tumors, there are 3 genes that are mutated, somatically mutated in more than 10% of cases. And one of them is GATA3. The mutations in GATA3 are almost always ER-positive breast cancer. And the mutations tend to fall in two clusters, not perfectly, but it's pretty close. So you get mutations that fall into this zinc finger domain, and then you get about half the mutations will fall at the C-terminus. And the mutations that occur at the C-terminus will almost always cause a single base pair insertion. So you get a frame shift into the +1 frame. And if you push the gene into the +1 frame, it will read through and hit the next stop codon that's in the +1 frame, which is 65 amino acids downstream. So you end up creating this protein, this GATA3, with this extra 65 amino acids of who knows what. Now, we've done the experiment. It's stable. Now, I put money on, actually, I put a chocolate bar in this in my lab and said, it's not going to be stable, it'll get degraded. And it turns out I was wrong. It is stable. So we've modeled it. We've tried to see what this mutation is doing. So we've used CRISPR to introduce a specific +1 frame shift in GATA3 at one of the sites we see in patients. It was not trivial. Now, people will tell you that CRISPR engineering is easy. It is not. If you're knocking a gene down, absolutely. If you're trying to do gene editing, it is very, very challenging. Stacey Jameson in my lab screened, at the moment she's screened 250 clones, and she's got 1 positive. Now, I'm going to show you the data from the 1 positive. The caveat is it's 1. Right? We've got 1 clone at the moment. It's not lack of effort. We're doing follow-up validation at the moment. But if we grow this 1 clone of mutant GATA3 with this extra 65 amino acid tail as xenografts, they grow slow. They grow really, really slow. These tumors never really take off. We see this here in vivo, but we also see it in vitro when we do the cell culture experiments in the lab. And this is a little bit surprising for us, but actually if you look at the survival data, so if we stratify patients based on their mutant GATA3 status, so the red is tumors with mutant GATA3, and the other line is wild type, you can see actually these patients have a better outcome. This is a little surprising to me, and it was as well to a lab I met in Japan recently, because they've been looking for this mutation using circulating DNA, and they have looked in many, many cancers, and they've never seen it. And they thought it was an artifact or a technical issue, and I don't think it is. I think these mutations are protective. And actually, you're not going to see them in metastases, because women that have this particular mutation in GATA3 tend to have a better outcome and are less likely to metastasize. And this raises this question. Why would a mutation that confers better outcome be enriched in cancer at the rate it is? And I'm struggling to find an example of this, and I can't get my head around it, but the data is the data, right? And so what we've done is we've gone and we've mapped the ER binding in these mutant cells or the wild type cells, and I wasn't sure what to expect. I thought we were just going to perturb estrogen receptor binding, and we do, but we also reprogram estrogen receptor to a bunch of new sites, and you can see that here. So there are thousands and thousands of ER binding sites that we only see in the presence of this particular GATA3 mutation. And you can see this here in the GATA3 ChIP-seq. They are sites that GATA3 binds to, FOXA1 binds to, and they're transcriptionally active as indicated by this histone mark. So this mutant GATA3 is actually pushing estrogen receptor around the genome to sites. We don't know what these sites are. We don't know if they overlap with the good outcome sites. We're doing that experiment at the moment, but we know they're transcriptionally active. And this, of course, is changing the target genes that the estrogen receptor complex is switching on and switching off. And this, of course, is likely to affect endocrine responsiveness. So finally, I want to thank the people that did the lab, people in the lab that did the work, particularly Hisham Mohammed, who set up RHYME and did all the work on PR, Karen Rossinas, who set up the ChIP-seq and the clinical samples, Sanjeev Kumar, who is the clinical fellow in my lab, who's going to be running the progesterone clinical trial. We've had really, really good support from Carlos Caldas' lab and his fantastic team of people. The drug screen, which I didn't really talk about, was done in collaboration with CRT, and we've had really, really long-standing and fantastic relationships with Wayne Tilley and his lab. So thank you very much.
That was a fantastic talk, and it was a superb example of how really outstanding basic research can lead to very important translation and potentially future therapies, improving therapy and treatment. So it gives me great pleasure to present the Society for Endocrinology Medal for 2016 to Professor Jason Carroll.
Excellent. Thank you very much. Thank you.
Congratulations.
Thank you. Thank you. Excellent. Thank you.